So the microscope is basic information. So you will have a virtual lab on looking at certain things. Let's see, the virtual lab for today is, again, pretty cool. I've done it before. It's as far as the microscope goes, it's on, let me see. Where did it go? We'll be doing how to operate the microscope and looking at animal cells. So you'll get a little exposure to the cell under a microscope. So you'll do that virtual lab this week or today, whenever you do it. So enjoy yourself. This week is starting next week. The labs get a little more timely and a little more involved. So let's go to the next one. So this is the way you hold the microscope. Look at this right here is the arm. So there's one hand on the arm and one hand on the base. And these are pretty close to the microscopes they have in the labs over in Valhalla. And here's the part. So here you go. Sometimes there's only one ocular lens. Like these are called ocular lenses, and they're for your eyes. But this is a binocular, so it has two ocular lenses, and they're 10x. So whenever you're doing a magnification with the objective lenses, you always have to add that 10x, which you're going to learn that today in the lab. This is the head, which sometimes rotates to the other side, like if the Finizio is over here with his bald head and he wants to see what you're looking at, he could turn this around and look at your specimen or your slide. There's a nose piece that kind of rotates, and that's where the objective lenses are. So I like to use the lab may tell you different, but I like to use 4x as a scanning power. So you can see what you're looking at under low magnification, very low magnification. So you just get an idea of what you're looking at. Then there's a 10x, which 10x, not 100x. 10x is, I like to call that low power. And Image Connect may call this 4x low power, but I like this better. Scanning, low power, and then 40x is high power. Now, in the lab, we don't usually use 100x too much because it's oil immersion, but you'll use it today probably. So you use oil as a droplet on the lens. And it's the longest lens too, like the 4x is the shortest, usually corresponds with the color. Then 10x is the next longest, and then longer still is the 40x, higher power, and then 100x is really long. So you start always at 4x, and you should always put the microscope back on low power, on scanning power, on 4x. Put it away on the shortest lens. So nobody breaks the slide when they put it in there. And it's easier for the next person. So that always should start on 4x scanning power when you put it away and when you start. Yeah, so there's a power too. There's a power cord. You plug it in. There's a power switch. And just like some lights in your house or lamps, that the power switch also controls the brightness of the light. And you're going to take your microscope slide and you're going to put it right here between the clips. And this one kind of moves back so you can fit the clip in nice and tightly and level. And then you can control the stage, and the stage can move like this way, forward and back, and side to side, based on two little handles here. So that, the light's coming up from the bottom, into the objective lenses. So you could pretty much put the light right through something you see on the slide with the naked eye. Sometimes you can't see anything because it's so small, but the specimens that we would look at, you could see them on the slide with the naked eye. And then once you're at 4x, which this one is on, 4x is scanning power. And you use the coarse adjustment first. Always use the coarse adjustment first, and that lifts the stage up and down if you need it. So you want to focus on 4x, scanning power, on coarse. And then you use the fine for your vision to make sure you could see it, or somebody else would use only the fine. So once the coarse adjustment is in place and you could see what you're looking at that power, scanning power, using the fine focus, then you can move to the next power. And you shouldn't have to use the coarse adjustment when you move from 4x to 10x to 40x. All you should have to do is use the fine adjustment to see what you're looking at. Okay, so there's also something called a diaphragm here that kind of converges the light and a condenser that basically does the same thing. And you'll learn that today with the bright microscope, compound microscope. And by the way, this is called a compound light microscope. Probably one you've already used in high school or in college, whatever. So it's compound because it has all these different objective lenses. You can change the powers, and light because it has a nice light source. It's not a dissecting microscope, which has a stage where you're looking at like a fly and you're dissecting a fly or something, or some small specimen. And it's a dissecting light microscope, but this is a compound light microscope. And they're much stronger. You have the transmission electron microscope and the scanning electron microscope, which are very strong, high magnification. I think TEM is the most magnification you can get. Scanning electron is the one in between. So this is the close-up of how the slide will fit in there. And you should label the slide, but this could be an exam, right? Where it's an unknown and you have to decide, is this epithelial cells like cuboidal cells, or is it connective tissue, areolar connective tissue? So it'll be set for you, but this is the way it clips in. This is a movable snapping back, and it's on the stage, and you adjust the stage position. So here's all the definitions of everything, and you should know pretty much. They're pretty easy, right? We talked about the arm, the base, the condenser, the coarse adjustment. Depth of field is like what you see. Like we would practice. We'd put like a slide on the microscope, and it has like threads on top of each other in different colors. So you can see, like, do you see the one that's closer to the objective lens clearer or the one further away? So again, you can't do that, but you'll get to do it in the virtual lab, hopefully. Then you have your adjustment ring. You'll see what that is. Diopter, this one doesn't have that. Your eyepieces are part of your objective lenses. Field of view is kind of important because that once you get into the right magnification, like say you're looking at blood. I think that's behind me right now. That's a blood smear behind me. So in your field of view at that magnification, you might say, well, I can see a lot of red blood cells, but I only see 2 white blood cells in this field of view, which is good, which means that if you don't have a lot of white blood cells there, that means that there's no infection in that blood. So field of view is important, and you can measure that in micrometers or millimeters on some microscopes. The fine adjustment knob is for your vision. So the course should be set once you move from scanning power and above, and then you just need to adjust the fine adjustment. The head, you saw where that was, the iris, the light control with the on and off, the mechanical stage that can move to and fro or side to side, then the stage controls there, the nose piece, which is where you carry it, nose and base. I'm sorry, nose piece is up top, the objective lens. The arm is the part that you carry it from. Objective lenses are the different lenses at different magnifications, 4x, 10x, 40x, 100x. Power switch, which a lot of times is also light intensity controller. And then the illuminator, which comes up from the bottom, from the base up into the objective lens. And the working distance, which is the distance between the mechanical stage and the objective lens. So that's why, like, you want to keep that distance clear. Like, you don't want to, go to 100x because you might break the microscope slide, right? The glass microscope slide. If you bring down the objective lens into the slide, it'll crack. So that's why you should always start with the scanning. And in our labs, we really don't go over 40x for that reason, unless we want to use oil immersion, then we're really careful. Like in general bio, we do that. Right. So here's some troubleshooting, which you'll probably go through. It shows you that working distance so you don't bust the lens. Bust the slide. Right there, this is a 4x. So you always start at 4x, which is scanning power. And you can measure. Some microscopes have a ruler, but in our labs, we actually put a little ruler on the slide itself so you can measure your specimen. And you can convert that metrically. Like this is inches. We don't use inches, but we can look at the one in centimeters. So there's 10 millimeters, right, in 1 meter. So you do the conversions, right? Millimeters to centimeters, but this usually has millimeters on it too. So you can see the millimeter measurements. Something really small like a blood cell, of course, is like a micrometer. It's a lot smaller. So you'll learn how to measure, get the total magnification. So in the virtual lab today, it'll show you, you have to multiply the 10x of the objective lens times the 10x of the ocular lens times the magnification of the objective lens, which is variable. So you might look at field of vision, I'm not sure, but whatever's in there, we'll talk about on the exam. So this is what we talked about, depth of field. Like these are a bunch of cells on top of each other, like stratified cells. So the cover slip is up a little closer to the objective lens, because the objective lens is up here, looking downward, right? Your vision, the light's coming up through the specimen, and your eyes are up here, right, on the objective lenses. So we'll probably go through depth of lenses. And here's another picture of a dissecting microscope. This is a dissecting microscope, which has that stage plate where you put the specimen on here, and you can experiment and see and take pictures. You know, some microscopes you can hook right up to your computer and get it up computer screen and save it and use it. So again, this is not an in-class in-person lab, so you're gonna have to rely on these pictures and everything else. So you can test yourself with this one. This is helpful for the exam, go through this, but again, you're gonna do the virtual lab. So get that done, and that is it.